Hi, I'm Mike. This is my presentation on Jennifer Doudna. Now, why did I choose her? Well, Doudna was one of the two women that are credited with the discovery of CRISPR-Cas9 in 2012. CRISPR-Cas9 is what I believe to be one of the most important technologies in medical history. Well, at least it has the potential to be. And I wanted to know more about the scientists that led to this new avenue of treatment. Now, Doudna was raised in Hawaii and developed a fondness and interest for the plant and animal life on the islands. Because, I mean, how can you not? It's Hawaii, it's tropical stuff, it's beautiful. Her father kept many books in her house about science. He was an English professor. And she was influenced by these books, but especially the one, The Double Helix, on the discovery of DNA. She went to school and earned her BA in biochemistry in 1985 from Pomona College, and she later earned her PhD in biological chemistry and molecular pharmacology from Harvard in 1989. Before her work on CRISPR, Doudna worked on research for ribosome structure and function. During her work, she found that the lack of visibility for the molecular mechanisms of the ribosome was really problematic because they couldn't really see what was happening, and so she went to go study for that exact thing. She used some science that I don't really understand of X-ray diffraction-based structure of the active site of the ribosome. And this is a rough sketch of what they ended up finding. And she continued to work on that at Yale until 2009. Now, CRISPR-Cas9 is where it really gets interesting and where she spent most of her time in the scientific community. With the help of, there's a French name, so I'm going to butcher it, Emmanuel Charpentier, Doudna discovered that the immune system protein Cas9, which defends the bacteria Stereococcus by attacking the DNA of its targets, could be reprogrammed with RNA to attack specific gene sequences. And this is huge because if you've ever looked at a DNA sequence, they're huge. But being able to program for a specific one allows for very quick and very accurate editing of genes. And it has a potential to be the most important gene therapy tool we've ever come up with. And it's already been further developed by other teams to treat things like sickle cell anemia, cystic fibrosis, Huntington's disease, and HIV, with varying success. Sickle cell anemia has been the most successful one so far. Doudna and Charpentier earned the Nobel Prize this year for their discovery of Cas9 in 2012. And this is significant for two reasons. One, it's one of the fastest turnarounds for a discovery prize, only further solidifying how important this tool is. And it's also the first Nobel Prize in science ever won by two women. That seems like it should have been a while ago, but it's huge that it happened at all. And they've run into some difficulties because Doudna and Charpentier only kind of described how CRISPR-Cas9 could be used for gene editing and weren't actually the first people to do it. So they've been battling for the patent for the technology with the team that actually did end up doing it and writing a paper on that. And what should you take away from this? Obviously, I want you to look into CRISPR-Cas9 because it's the coolest thing ever. But I also want you to think about how interesting it is just to be living in the same world as people like this. Like, people can look at these sort of DNA and like immune system defenses and be like, oh yeah, that can edit DNA. That makes sense. But I also want you to think about how the scientific community doesn't really care who you are. If you are smart and you discover something, props to you. I'd love to see more women in science. I'd love to see more people in science for that matter. Thank you.